Cell technology and cell cycle. Biological solutions and molecules can be studied through the use of a spectrophotometer. Light, ranging from near infrared to visible to ultraviolet, is split into its component wavelengths or colors by a prism or diffraction grating. Individual wavelengths are selected and passed through the sample solution. This is repeated for the other wavelengths. After leaving the solution, the intensity of each wavelength is measured. The intensity of the various wavelengths enables the scientist to discover information about biological solutions and molecules. This is particularly useful in studying DNA, RNA, proteins, and enzymes, as they all have different light absorption properties.
Microscopy. Many organisms that are key to biology are very small and can only be analyzed through microscopes. Conventional microscopes can resolve structures as small as 200 nanometers or 200 times 10 to the minus 9 meters. Electron microscopes that use a beam of electrons can resolve structures as small as 0.1 nanometers. Shown below is an electron microscope picture of C. elegans cells, or roundworm cells. DNA sequencing. DNA sequencing discovers the order of nucleotides in a DNA molecule by using multiple laboratory techniques and extensive computing resources. Basic research is used to identify a gene of interest. The gene that is to be studied or modified is then isolated by using restriction enzymes to cut out the gene from the chromosome. Computers attached to robotic DNA sequencers fragment, analyze, and find a gene based on user input. Gel electrophoresis. Gel electrophoresis is used to separate DNA and RNA fragments and protein molecules based on their electric charge and size. The DNA fragments are stained and then placed in a gel with collagen fibers in an electric field generated by a positive and negative electrode. DNA has a negative charge, so it is attracted to the positive electrode and moves through the gel towards it. Smaller pieces of DNA can move through the gel quicker than the longer pieces that get tangled in the collagen fibers. This enables the smaller pieces to move farther in the gel. The fragments are separated according to their size and electric charge in a banding pattern. Before electrophoresis, it used to be very difficult to find specific genes on DNA. Polymerase chain reaction. Once the gene is isolated, copies need to be made in the amplification process. PCR, polymerase chain reaction, was designed to make billions of identical copies of a gene within a few hours, making the gene easier to study and to use in genetic engineering. This reaction is carried out by a special machine that utilizes repeating cycles of heat, DNA polymerase, DNA primers, and free nucleotides to build copies of the DNA fragment. The techniques of human gene sequencing were used to achieve one of the most spectacular biomedical achievements in history. The entire genome of humans was mapped. The correct order and placement of the 4 bases, adenine, thymine, cytosine, and guanine, that make up the DNA was found. The project was started in 1990, and in 2003, 92% of the genome was mapped. New gene sequencing technology was developed, and then 100% of the genome was mapped. The genome information has already led to pharmaceutical and biotechnology advances, including new medicines and treatments for genetic disorders. As the project was distributed over 6 countries and 20 institutions, it is hard to estimate the total cost, but it is approximated as $2.7 billion. Human gene therapy. Gene therapy involves the modification or altering of the expression of a gene to change the biological properties of cells to improve the health of the patient. Categories of gene therapy include replacing a gene that is causing a disease with one that is healthy, inactivating a disease-causing gene, or modifying a gene to create a new one that has positive benefits. Here are some examples of some successful gene therapies: sequencing the genomes of tumors to identify their mutations and treating patients with targeted treatments based on the findings, using cancer patients' own T cells to fight cancer by stimulating them to increase their number in the lab and then returning them to the patient's blood, developing safe, highly effective vaccines to prevent infections by pathogenic microbes, treating human diseases with proteins made by microorganisms. Insulin is the protein hormone that regulates sugar levels in the bloodstream and treats diabetes. The gene for insulin was found, isolated, and put into the bacterial chromosome of E. coli. The bacteria was able to produce human insulin very rapidly, which is commercially available for human use. Stem cells. In mammals, there are two types of stem cells. Embryonic stem cells are found in the blastocysts of developing embryos. Embryonic stem cells can differentiate into any type of cell. They are known as pluripotent because they can differentiate into any cell type present in the organism. Adult stem cells act as a repair system by maintaining the turnover of regenerative organs such as blood, skin, or intestinal tissue. They are known as multipotent because they can differentiate into some, but not all cell types present in the adult organism. Embryonic stem cell technology. Presently, embryonic stem cells have been used primarily for research. Potential for technologies exist, but currently no product has been produced. The use of embryonic stem cells is ethically and politically controversial. Promising research using embryonic stem cells are things like tissue engineering for organ transplants, in vitro models to test drug response and predict toxicity, the creation of neurons for the treatment of Parkinson's disease, and alternative treatments for diabetes. Cloning. Cloning is the process of producing individual organisms with identical or virtually identical DNA, either by natural or artificial means. In reproductive cloning, researchers remove mature somatic cell, such as a skin cell, from an animal that they wish to copy. They then transfer the DNA of the donor animal's somatic cell into an egg cell or oocyte that has had its own DNA-containing nucleus removed. The egg is allowed to develop into an early-stage embryo in the test tube and then is implanted into the womb of an adult female animal. The adult female gives birth to an animal that has the same genetic makeup as the animal that donated the somatic cell. This young animal is referred to as a clone. Reproductive cloning may require the use of a surrogate mother to allow development of the cloned embryo. Transgenic organisms. Transgenic organisms are created by genetic engineering techniques where a genetic sequence is transferred from one organism into another with the help of certain bacteria. The goal is to create an organism that is stronger, healthier, and more disease-resistant by taking advantage of a gene from another organism that has those traits. The desired gene is cut out by restriction enzymes and then isolated using the process of electrophoresis. PCR is then used to make billions of copies of the gene. The desired gene copies are combined with opened plasmids and carry out millions of ligation reactions to join them, producing a mixture of plasmids with added genes, some useful and some not. The bacterial colony that carries the desired gene is placed in a large liquid media culture, greatly increasing the number of bacteria cells as they reproduce in the favorable conditions. The bacterial cells containing the desired gene are placed in a container with the cells of the target organism. While it is a rare event, if enough copies of the bacterial cell are present, some of the target organism cells will take up the introduced DNA and are called transgenic cells. The nuclei of fertilized embryos from the target organism are isolated, and the transgenic cell nuclei replace the embryo nuclei, creating transgenic embryos. A transgenic embryo is implanted into the organism's uterus, and the child will then be transgenic. Genetically modified crops. The genetic engineering approaches to make transgenic animals are similar to the methods used to make genetically modified GM crops. Genes from other organisms can be introduced into crops to improve their nutrition content, make them resistant to pests, extreme temperatures, drought, and high salt, and make them less dependent on nitrogen fertilizer. Eukaryotic cell cycle. The eukaryotic cell cycle has two major divisions, interphase and the mitotic phase, which you can see in the graphic. Interphase is this big, long phase in blue, and then the mitotic phase in green. During interphase, the cell metabolic activity is very high. It is busy growing and copying its DNA and organelles so it can divide. The mitotic phase includes two events, the division of the cell's nucleus, or mitosis, and then the division of the cell's cytoplasm, cytokinesis. Mitosis involves a series of steps, or subphases.
Most cells spend more than 90% of their lifetime, their cell cycle, in interphase. Interphase has three distinct subphases. Gap 1, where the cell increases in size and its supply of proteins, particularly those used in the duplication process. Synthesis, where DNA replication occurs, doubling the number of chromosomes. At this point in synthesis, or S phase, chromosomes cannot be seen individually because they have not condensed. And gap 2 is where cells go in preparation for cell division. The cell completes its growth, checks its DNA for any damage that might have occurred during replication, and ensures that all required proteins are present. Mitotic phase. After a cell completes its preparation for division, it enters the mitotic phase. The mitotic phase has two phases. Mitosis, which is the division of the nucleus, containing subphases prophase, prometaphase, metaphase, anaphase, and telophase. And then the second phase of the mitotic phase is cytokinesis, which is division of the cytoplasm, which differs for plants and animal cells. Here are the phases of mitosis, and the previous slide shows them on a visual if you want to refer back to that. In prophase, the nucleolus disappears, chromatin condenses into chromosomes and appear as two sister chromatids joined at their centromeres. Centrosomes start to travel to opposite ends of the cell, and spindles start to form from the centrosomes. In prometaphase, the nuclear membrane disappears. The spindle is nearly complete, and chromosomes attach to the spindle at their kinetochores. During metaphase, the spindle is completely formed. Chromosomes align on the metaphase plate, or the equator of the cell. Anaphase. Sister chromatids separate from each other at the centromere and are pulled to the two poles by the spindle fibers. Telophase. Cell elongation continues. The nuclear envelope around the chromosomes reappear, as do the nucleoli. Cytokinesis in animal cells. First, a ring of microfilaments forms a contractile ring around the outside of the cell. The ring forms a cleavage furrow, which splits the cytoplasm in two, and you can see that beginning to form here. Cytokinesis in plant cells is slightly different than in animal cells. Vesicles containing cell wall material collect in the center of the cell and then fuse together. The cell plate forms from the inside out and turns into a wall between the two new cells. The membranes surrounding the vesicles fuse to form new parts of the plasma membrane. Meiosis. Meiosis reduces chromosome numbers in diploid organisms to create gametes or sex cells. Like mitosis, meiosis is begun by a single duplication of chromosomes. Unlike mitosis, the overall result of meiosis is 4 daughter cells, each with half the number of chromosomes. They are now haploid. Sexual life cycles alternate between haploid and diploid phases. The fusion of haploid gametes during fertilization results in diploid offspring. There are 2 consecutive divisions, meiosis I and meiosis II. Here are the phases of meiosis I. In prophase I, the nuclear envelope disappears and the spindle begins to form. Homologous chromosomes replicated during interphase pair to form a tetrad. It is at this point that crossing over occurs. Metaphase I is when the tetrads line up at the cell equator, and the manner in which they line up gives each cell a unique combination of genes from each parent's chromosomes, known as independent assortment. Crossing over and independent assortment account for most of the genetic variation in populations. Anaphase I is where the tetrad splits and inherited chromosomes move to one pole or the other, independent of each other. In telophase I, the nuclear envelope reforms and the nucleus is now haploid. Interkinesis is the division of the cytoplasm, similar to cytokinesis in mitosis. The cells are now haploid. Meiosis II phases. Meiosis II follows the same steps as mitosis, but they are named prophase II, metaphase II, anaphase II, telophase II, and cytokinesis instead of interkinesis. Meiosis I starts with 1 diploid cell and finishes with 2 haploid daughter cells. Meiosis II starts with those 2 cells and finishes with 4 haploid cells in a process similar to mitosis. Cell cycle checkpoints. The cell cycle control system checks cells to ensure that important processes, such as DNA replication and repair, are working properly before division. It ensures that damaged cells are not allowed to divide. 3 major checkpoints exist to regulate the cell cycle. At the end of gap 1, there's the restriction point. At the gap 2, the G2 to M transition, so gap 2 to mitosis transition. And during mitosis, there's something known as the spindle assembly checkpoint. At each checkpoint, if cellular surveillance systems detect a chromosomal defect or a metabolic problem, the checkpoint is activated. Once activated, the cell will stop in the cell cycle and repair processes begin. If repaired, the cell is released from the checkpoint and continues through the cell cycle. If the cell cannot be repaired, a death program is initiated and the cell dies rapidly. G0 phase. Fully mature functional cells leave the cell cycle and carry out their specialized functions in G0 phase. If they re-enter the cell cycle, they will pass through the G1 checkpoint and into S phase. Examples of cells that are almost always in G0 in adults are neurons and muscle cells. Cells that are not ready to divide or have a defect should get stopped and repaired at gap 1, gap 2, or during mitosis. Uncontrolled cell growth results if a defective cell is not stopped, is not repaired, and does not die, leading to cancer.